Let us talk about the different types of cell divisions now. Cell division was first described by two scientists, Prevost and Dumas, in the zygote of frog. So they were the first ones to actually give this description of the cell division, Prevost and Dumas. Another scientist, Nageli, gave a concept that these cells are formed from other cells, which was confirmed and backed by Rudolf Virchow. He said that the cells arise from pre-existing cells. We have written about it, we have talked about it in cell cycle, that is omnis cellula acellula. That means cells arise from pre-existing cells. This original concept was given by Nageli, but it was confirmed by Rudolf Virchow. So the credit of giving this phrase that the cells arise from pre-existing cells goes to Rudolf Virchow. But Nageli was the first to give this concept. So this is about cell division. Three types of cell divisions that are which we talk of. Types of cell division. First is amitosis, which is also known as direct division. Direct division. Then the next is mitosis, which is known as indirect or equational division. Indirect or equational division. Equational because the two daughter cells are qualitatively as well as quantitatively similar to the parent cell. And 3 is meiosis, which is known as reductional division because the daughter cells get half the number of chromosomes as compared to the parent cell. So it is reductional division. So 3 types of cell division. Let us take amitosis first. In amitosis, the spindle fiber formation or disappearance of nuclear membrane, all these things do not take place. The examples where we find amitosis, one is amoeba. Certain cells of our body also show amitosis, like cartilage cells, that is chondrocytes. Diseased cells also show same type of division. What happens in this division, as we have taken amoeba as an example, so say this amoeba has to divide and there is a central nucleus. This nucleus, it becomes little elongated and shows a constriction in the middle. There is a constriction that appears in the middle. This constriction will deepen, that means from here it will go in and in, so ultimately it is going to go closer and it will divide into 2 daughter nuclei. So here now this nucleus has almost divided but still attached at a point. And at the same time we also start seeing the cytoplasmic constriction. So from here the cytoplasm also deepens, it goes inner, and the point is same for the nucleus as well as for the cytoplasm. So basically it is, you know, pinching. So you press it from the middle and then it breaks into two parts. So after this, the two nuclei would separate, and by this time the cytoplasm is also almost divided into two parts. And after this, these two will separate, each one will having its nucleus, and these two cells which are formed, they are known as daughter cells. In case of amoeba, they are called daughter amoebae. So what has happened is nuclear membrane is as it is, no spindle fibers, chromosomes are in the form of fibers, chromatin fiber. They simply, the nucleus simply pinches into two parts. It is like a balloon, if we press it in the middle, you start seeing two parts and then separated. So same thing happens, followed by the cytoplasmic constriction. This constriction deepens and finally it completes in the middle, forming two daughter cells. The disadvantage of this division is that invariably the distribution of chromosome is not 50-50. It could be 49-51 because, you know, you're pressing it from the middle. To be very precise, it has to happen in a particular manner. But if you just pinch it in, chances are that one part gets more number of chromosomes, other part gets less, which results later on, after few generations, it results into metabolic disorders. And that is why here we said some diseased cells show this kind. So probably it is this kind of division which is responsible for those diseases to occur in the body. So this is amitosis. In some cases it is also reported in yeast. Sometimes yeast also shows same type of division. So it is a very simple division. The reason why we are calling it direct or direct division is the nucleus simply pinches into two parts, that's it. So no specialized structures like mitotic apparatus or spindle fibers, nothing is visible. Simple constriction followed by cytoplasmic constriction to give two daughter cells. This is amitosis. Mitosis, which is known as indirect or equational division, is a little complex process. It is systematic for proper equal distribution of chromosomes. So let us take mitosis now. Let us see the various stages of mitosis now. Mitosis was explained by two scientists, Strasburger and Strasburger explained these mitotic divisions in plants or plant cells, and Fleming explained mitotic divisions in animal cells. The term mitosis was also given by Fleming. Term was also by Fleming. Mitosis is completed in two steps. That is karyokinesis. Karyokinesis means division of the nucleus or karyon, followed by cytokinesis, that is division of the cytoplasm. Karyokinesis is completed in four subphases: prophase, metaphase, anaphase and telophase. So 4 subphases of karyokinesis. This will complete nuclear division, and after nuclear division cytoplasm will divide. Now let us see, we are starting with mitosis in an animal cell, and after we complete this we will see how it is different in plant cells. The first cell that we are drawing is the interphase cell. Interphase cell means it has completed interphase of cell cycle. It is ready for M phase or mitotic phase. In interphase cell, the nucleus is large because the DNA content is doubled, cytoplasm is more, proteins are more, so it is larger in size. Whenever we draw diagrams of cell division, we are not really worried about the typical structure like double membrane, nuclear membrane, double layered, ER, Golgi. We don't draw anything. Because it is an animal cell, we will draw the centrosome. That is 2 centrioles and the 9 fibers around them, centrosome complete, and the nucleus. Plasma membrane, nothing else is drawn. So this is interphase cell. It is ready for division. Normally whenever we draw, the labeling is also not done. The diagram itself should be self-explanatory. There are some stages where we would be labeling certain things. To keep a track of number of chromosomes, we will start with a fixed number of chromosomes. And here we are going to see the threads. It is chromatin fibers. So 1, 2, 3 and 4. Say the interphase cell has 2n number as 4. The diploid chromosomes here are 4. This is only for our understanding. We can take 8, 10, 20, whatever. 4 is because it is easier to draw and easier to see. What does this mean? That it has 2n number 4, but we said it has passed through S phase. So there is actually 2n plus 2n. So with these chromosomes, there is one more, which is a copy. So during S phase, each chromosome made its copy and these are attached here. Here we have not shown chromosome, we are just showing the fibers. So these are chromatin fibers. 2n number is 4. Original 4. In S phase, all 4 made DNA doubled. So now each chromosome has a copy of it. So actually speaking, it is 4n. That is what happened in S phase. Now this cell enters the first subphase of karyokinesis. Now in all these phases, there is no clear-cut demarcation. There is no line that prophase ends here and metaphase starts here. There is a little overlapping which takes place. Prophase is a longer phase in case of or as compared to others. So we can draw early prophase and late prophase. For all others, we may not do that. So let us talk about the first one, that is prophase. And we are drawing 2 prophases. One is the early stage and then late prophase. The nuclear membrane, it remains as it is. Centrosome disappears. That means the clear cytoplasmic area around it disappears. The centrioles split and one pair of centriole would remain here and one pair after splits, it starts moving towards the other pole. That means the other one would start moving here. And from each centriole arise thread-like structures. These thread-like structures are known as asters. So or aster fibers. So these threads which we have drawn here, they are called aster fibers. And this complete structure, that is centriole in the middle surrounded by all aster fibers, this whole thing is known as an aster. This has happened in early prophase, formation of aster. What is the situation of the chromatin fibers? They have started to condense. Condense is that they lose water, they become compact, the nucleosomes which are there, they come closer. So instead of a fiber, we start seeing chromosome. It is not very distinctly visible, but yes, you can say that now there is a chromosome. So here there is a chromosome, and we will keep a track of the number. We started with 2n number as 4. So these are the 4 chromosomes, and attached to each one is the chromatid of the other chromosome. So instead of threads, now they assemble, they condense. So now we start seeing chromosome-like structure. Two distinct changes which take place in the early part of prophase. Aster formation. How is aster formed? The centriole splits, one pair remains at one end, the other pair starts to move at the other pole. Small fibers, they appear around these centriole, these are known as aster fibers. Centriole along with these fibers is known as aster. So there are 2 asters which are formed in case of an animal cell. Because it is an animal cell, we are talking of centriole. If it was a plant cell, centrioles are absent in plant cells. Now what would happen in the later part of prophase? So let us talk of prophase late. So early prophase and late prophase. In the late prophase, the nuclear membrane, it dissociates, it breaks up and joins ER. So those fragments of nuclear membrane will start to break off and they will go and bind with endoplasmic reticulum. This is seen as disappearance of this membrane. We call it that nuclear membrane disappears, but it does not disappear by magic. It disappears because these nuclear membrane fragments, they break off and they form the ER, they join ER. So now what is visible is the 2 asters, one at one pole, the other at the other pole, the aster rays, and there are some more fibers which appear between these 2 centrioles. So these fibers, they extend. And because of this typical shape, it is known as spindle apparatus. These fibers are called spindle fibers. Two asters and spindle fibers together, known as mitotic apparatus. We will draw mitotic apparatus separately. Because the nuclear membrane is now gone, it has dissociated, disappeared. What is the position of these chromosomes now? The chromosomes come to lie in the cytoplasm. Now they are free in the cytoplasm. So let us draw these chromosomes, and we can draw them anywhere. And we have to draw the copies with them. So this complete spindle apparatus appears towards the end of prophase. In the beginning, the chromosomes start to become thicker due to losing of or loss of water. Towards the end, these chromosomes come to lie in the cytoplasm as nuclear membrane is completely gone. and from the centriole you start seeing some fibers which are running from one pole to another. Now these chromosomes which are here, they will arrange themselves on a plane. If these two are considered as the poles, the middle line becomes the equator. Similar to our earth, there is no such line which goes there. So this is an imaginary line. So here we are going to imagine that there is a line, we will call it equatorial plane, and it will enter the next phase, that is metaphase. So after prophase, the cell enters metaphase. In metaphase also we can show early and late, but everything we will represent in one stage only. All chromosomes, they appear in the equatorial plane. Let us finish these basic things which are there, the asters, the aster rays, and the spindle fibers. The spindle fibers, aster rays, they are made up of tubulin proteins. So this is of tubulin protein. These are microtubules. We have studied the structure of microtubules. So these spindle fibers are made up of the same microtubule. Now these chromosomes, they are thickened further and they will arrange themselves on the equatorial plane in such a manner that one chromosome faces the upper pole and the other chromosome faces the lower pole. So if there are two chromosomes, they would be placed like this, one towards one pole, other towards the other pole, because they have to get separated later on. So the arrangement is going to be one chromosome, and see we are drawing them little thick here, and its copy, this is one chromosome, the second chromosome, these two are fused at the centromere. Third chromosome, its copy, and fourth chromosome. Four. But how do we show them on the equatorial plane? All of them should be here. So the attachment should be, I am making the spindle apparatus separately now, slightly bigger, so that we are able to see what exactly is the structure. This is one centriole, the second centriole, the aster fibers, the spindle fibers. Now the spindle fibers are of two types. One spindle fibers, they run from one centriole to another centriole. Then there are the second type. The second type are the ones to which these chromosomes are attached. So this is the centromere, this is one chromosome and its copy. Second chromosome, and its copy. Let us make the third one here, its copy, and the fourth one here, its copy. So all of them are on the equatorial plane. We see two types of fibers. Certain fibers are attached to the chromosome. How are they attached? This is the chromosome. This is the centromere, the chromatids of one and the chromatids of the other one. This is how the pair is. This centromere on its two sides has a little paddy structure which is known as kinetochore. The spindle fibers are attached to this. So they are attached to the kinetochore. So when the spindle fibers pull or contract, they'll pull these two chromosomes apart. Now in this mitotic apparatus, this is called mitotic apparatus. What are these structures that we are seeing here? We said this is aster, and there are two types of spindle fibers which we see. One which are connected to the chromosomes. These fibers are known as chromosomal fibers, and the others which run from one centriole to the other centriole, they are not connected to anything. These are known as continuous fibers or interpolar fibers. So the second one, this type, this one we are talking of. This one is interpolar or continuous fibers. Chromosomal fibers are not continuous. So from one end they are coming here, attaching to the kinetochore. From the other end also they are attaching to the kinetochore. So this is the spindle apparatus to which these chromosomes they would arrange on the equatorial plane. So this is mitotic apparatus which we see in metaphase. Now the cell enters the next phase. After metaphase, the cell enters anaphase. So next stage is anaphase stage. Again in anaphase we can make early anaphase and late, but we are drawing only one. So this is anaphase. What is going to happen in anaphase? These spindle fibers, they are going to contract. So when they contract, these two chromosomes, they will be pulled apart. So here let us draw this centriole with its fibers, that is complete aster. What is going to happen to the continuous fibers? The continuous fibers are going to remain as it is. But the fibers which are attached to the chromosomes, when they contract, the two chromosomes will be separated. And how will they get separated? They will get separated because the centromere here is going to split. So it is connected, joint, one centromere, and when these two chromosomes move apart, the centromere splits. So this becomes the centromere of one chromosome and the other becomes of the other. So there are these chromosomes which have moved apart. One is the original one and the other is the copy. Anyone can go on any side, so it is not fixed. We have used two colors, the yellow and the pink one, so we can show anywhere going on any side. But when they separate, we find a fiber between these two separating chromosomes. These fibers would appear only in anaphase. We don't see them in metaphase. These fibers are known as interzonal fibers. So in mitotic apparatus, if the question is asked only about draw the mitotic apparatus, we can draw it in this stage, that is metaphase stage, where we have to show two fibers. One is the continuous one and the others are the chromosomal one. And one we can show it in anaphase, where we start seeing the third fiber also, which is interzonal. Interzonal appears between the two separating chromosomes. The reason for formation of all these fibers? Or if we put this in a different manner, how are these chromosomes moving apart? What are those forces which are taking these chromosomes away from each other? There are two main things which are responsible for it. One, the chromosomal fibers, they contract. So when this contracts, it is going towards the pole, this is going towards the pole. So contraction of the chromosomal spindle fiber and extension of the interzonal. So this interzonal is increasing on both the sides. So the middle part is becoming longer and the end part is shrinking or contracting. So this pulls the two chromosomes apart. In anaphase, as we said, early and late stages can be drawn. Early would be when you have shown the chromosomes here. In late anaphase, the chromosomes would go even closer towards the poles. That is the only difference. So interzonal fibers will become even longer and chromosomal fibers will become shorter. That's the only change which we would see. After anaphase, the cell enters telophase. Telophase is actually reverse of prophase. Whatever happens in prophase, everything gets reversed in telophase. What happened in prophase? Nuclear membrane disappeared. So here the nuclear membrane will reappear. Again, it is not magic. Why did it disappear? Because the fragments of nuclear membrane, they break off and join endoplasmic reticulum. Here, those fragments of endoplasmic reticulum will break off and will join to form the nuclear membrane. So now here the nuclear membrane starts to reappear. How many nuclear membranes will reappear?